The last three slides in terms of photosynthesis and carbon fixation, we're going to talk about the synthesis of a couple unique sugars. And so we'll talk about first lactose, and then we'll talk about vitamin C. Lactose is a sugar that is produced after birth, and so the synthesis of lactose actually only occurs during a portion of the life cycle. And so what happens is, for the synthesis of glucose, what we're going to use is an enzyme that is going to be a galactose transferase. And normally in the body, what we will have is a galactose that has been activated by the use of UTP will be linked to, or covalent linkage will be made with a N-acetylglucosamine. And what this linkage between these two sugars to make a disaccharide is the beginning portion of a sugar that is normally displayed on the outside of cells. And what happens on the outside of cells that we'll learn about in the next chapter is we're going to place sugars in a particular order, place them on the outside of the cell as a signaling molecule. And so this is the beginning portion of where we're going to make a polymer, a short polymer that has three-dimensional structure, and it will end up being part of a glycoprotein. So that is the normal enzyme that is used, but we're going to modify how that enzyme works to actually synthesize glucose. What will happen is during the postpartum phase, we're going to have a protein that is produced called alpha-lactobumin. Now this alpha-lactobumin will bind to the transferase, and when that does, it manipulates and changes the substrate specificity for this enzyme. Now it will still use a galactose, but instead of binding what we had previously, the N-acetylglucosamine, where we have the acetyl group and an amine in the position where there was a hydroxide, we're now going to bind glucose. And so the specificity of the binding or the reactants with this combination is glucose and the UDP galactose. It'll facilitate UDP coming off and creating a linkage to create the sugar lactose. And this lactose will be produced for a short period of time. The next sugar that we're going to look at is the synthesis of ascorbic acid. And so in ascorbic acid, well, what we can call, most people don't know it by that name. Most people think about ascorbic acid as being vitamin C. This is an antioxidant, and you've probably heard quite a lot about vitamin C. And humans do not have the enzyme necessary to convert this last step, whereas in plants, this enzyme is available in plants to create the vitamin C. Now, why in the world do we show this pathway, and why do the mammalian systems actually have this type of synthetic process? Well, what happens is we found the utility of this particular molecule as being very important in biological systems. And what happens is this activated sugar, okay, that's activated because it has a UDP, is able to react with things that are exogenous or not supposed to be in the body. And so what we see with, let's say, someone that has taken a drug, one of the way that drugs work, most of the time they're very hydrophobic, and they only last a certain period of time. We clear drugs by either breaking them down, or we covalently modify them so that they can be degraded and or extracted. This particular molecule reacts quite often with those molecules that are not supposed to be in the body, and if it is particularly hydrophobic, adding a sugar of this nature, which has the acidic sugar on it, makes that molecule much more polar. The more polar it is, the more soluble it is in the bloodstream, and the more that it can be taken into the filter, into the kidneys to be filtered out. And so a lot of different material are linked to this particular sugar, and create the modifications that can be filtered out, including drugs, smoke products, all sorts of different organic molecules that get into the body. Moving on to chapter 13, we're going to talk a little bit more about carbohydrates and how they are utilized within the body. And so, what kinds of ways that we find carbohydrates in the body, because at this moment in time we've seen monosaccharides in terms of different sugars, glucose, galactose. We've also then seen a couple of ideas about sugars being disaccharides, especially in those that we take in as fuel. But the sugars are very important in a lot of different processes within the bodies. And so when we think of sugar, most of the time we think of absolutely energy sources where we break them down and degrade them to produce ATP in an aerobic form. But much more often, there are more items that sugars are used that are structural and informational that we sometimes kind of overlook. And so if we look at the structural framework of DNA, RNA, the ATP, the energy molecules, let's say the FAD, NAD, the electron carriers, if you go all the way back to their elemental structure, they have a 5-membered sugar called ribose as part of the framework, as part of the structural component. And so none of these entities would be available to the body unless we had the sugars available. Also, we have sugars that are incorporated elements as part of cell walls and exoskeletons. They actually can be very structural in nature, where they give the integrity and strength needed for structure. And then, lastly, there's a whole class of use of sugars to create information. And what will happen is we're going to place sugars in a particular sequence of bonding, which will create unique three-dimensional structures, and each three-dimensional structure can be informational, saying that I might put a certain sugar on the outside of a cell to distinguish that cell as being a certain type because of that shape that is on the outside. Sugars, by the virtue of being able to bond in many ways, let's say it has 6 carbons on each sugar, and so you could take 2 sugars and link them 1 to 1, alpha, beta. So 1 to 1 alpha, 1 to 1 beta. You can go 1 to 2 alpha, 1 to 2 beta. You can go 1 to 3 alpha, 1 to 3 beta. And just even 2 of the same sugars, and you go through all of those permeations, the rotation of that sugar, whether it's alpha or the beta, creates very unique three-dimensional structures which are different. And so the more sugars you add, you have more variations, and it can be a complex mechanism to create unique three-dimensional structures. Now, classes of carbohydrates, we've seen a couple of these: simple sugars, monosaccharides, disaccharides, and then something new called oligosaccharides are more than 2 but less than a very large number of saccharides. The number can be different between what books you talk about, and so oligosaccharides are mini saccharides, but you get kind of into a different form when you get to what's called polysaccharides. Polysaccharides are polymers of sugars. There is homopolymers, meaning the same sugar put all together and no other sugar there, and there is a linkage. These are storage molecules like starch and glycogen, and they can be structural like cellulose. And then you can have heteropolymers, in which the polymer is a collection of maybe disaccharides that are repeated, but very long chains. Oligosaccharides are relatively short. polymers, the polysaccharides are relatively long. Then we have what are called glycoconjugates, okay? And glycoconjugates are sugars attached to something, and so we'll have something called a glycoprotein. So it's a protein that has sugar on it. We can also have proteoglycans, a protein and a sugar. Well, a sugar protein and a protein sugar. I'll talk to you what's really the difference in that. In that the glycoprotein is mainly protein with a sugar that is on the outside, whereas a proteoglycan is mainly sugars with proteins incorporated in them. And then glycolipids, sugars that are attached to lipids. The glycoproteins and the glycolipids are intended almost always to be informational, and the orientation of these sugars are almost exclusively on the outside of cells. The proteoglycans tend to be ones that are released between cells, and we're going to get some thickening of the liquid, and we get some of the sponginess. You'll find these in joints and in the extracellular membrane matrix that will help the cells hold together. Glucose, normally the first thing that we think about is storage in terms of energy. In the animal form, there is glycogen, and in plant form, we have starch. There are two versions, amylose and amylopectin, and so we'll take a look and see what the differences are. Amylose is the most simple type of starch. It is a homopolymer of glucose. They are all alpha-1,4 bond linkages, and it is straight chained. What's interesting about the structure of these is because of the chair conformation, the structure of this amylose tends to spiral, and so that is indicative of the amylose. It can be degraded by amylase into maltose, maltotriose, and dextrin. Maltose is two sugars, two glucoses. Triose is 3. Dextrin is a name for at least an oligosaccharide or a polymer of just the glucose sugars in an alpha-1,4 matrix. The maltase can convert the first two, the maltose and the maltotriose, into glucose itself, and then dextrinase can convert the dextrin into glucose in its degradation. Amylopectin is mainly alpha-1,4 linkages. It doesn't spiral because, on average, between 24 and 30 sugars, there is an alpha-1,6 linkage. And so what this does is this link places one sugar above this chain, and then that can be alpha-1,4, and this branch point is relatively far away from each other. The enzyme that degrades this is amylase. And so we have alpha-1,4 for the main body, and every 24-30 we have a branch point. If you actually take a look at a glycogen, I didn't actually change the structure of the picture that I had. Alpha-1,4 for the main chain, and then an alpha-1,6 branch point. The difference between the amylopectin and the glycogen is that for glycogen, on average, there is 10 residues before branch point. So it's much more branched in glycogen than amylopectin. If we talk about the consequence of that in three-dimensional terms, the amylopectin does have branching, but it's much less branched, meaning that as they grow, they tend to grow in granules, where we have a core and then we grow out in all directions. The branch point will increase its solubility and will increase the number of ends that are accessible on the outside. Glycogen, because of its ability to branch much more often, what we can see is there are a lot more ends, and the ends are where degradation happens. And so you can talk about the structure kind of like a koosh ball. You have a koosh ball that has a few actual fingers, or a koosh ball with lots and lots of fingers. The glycogen that we have in our body looks like this that has lots and lots more ends by which we can manipulate the glycogen both degradatively and synthetically. This structure allows us to, one, create a much more soluble glycogen, and two, a more accessible glycogen. Now, if you actually look at glycogen under a light microscope, you see these dark spots. These are glycogen granules. They are large enough to see under a light microscope, and so they're relatively large. We find glycogen stores most often in liver and muscle, and the average molecular weights of these packets, these granules, are in the millions of grams per mole. What is interesting about these glycogen molecules is that the glycogen, its primer from which the glycogen grows, all of the enzymes for synthesis and all of the enzymes for degradation are continuously bound in one of these granules. Now, remember, in degradative and synthetic processes within the body, we never want to have both synthetic and degradative processes to occur, because that would be a waste of energy. We synthesize it and degrade it, synthesize and degrade it. That would get us nowhere. And so the manipulation of a glycogen granule, whether to synthesize it or to degrade it, requires a high level of coordinated regulation. We'll go ahead and look at that particular coordination in detail in the next chapter. Glucose as structure. So if we take a look at the polysaccharides, this is where we get structural components. And so again, cellulose, the single most synthesized biomolecule in the world, is the largest component of stalk, stems, and trunks, the woody portion. And so this, and if you think about wood, has a lot of integrity, has a lot of structural integrity. You wouldn't think that glucose by itself might be a very strong bond, but because of the structure of cellulose, which is different than the storage molecules, we get quite a bit of structure. Now, chitin, on the other hand, is a polysaccharide, not just having glucose. We're going to have some modified glucose molecules that will allow it to have more hydrogen bonds. The components of exoskeletons of insects, lobsters, crabs, we can see, and if we think about the structures of those particular components, a lot of structural integrity. And so this is the second most abundant polysaccharide found on the face of the earth. Cellulose, the only difference between cellulose and the storage molecules of glucose is the bonding. It is still a glucose between a 1 and a 4 bond, but it's in the beta conformation. And what that allows to have happen is that in the beta conformation, in the chair conformation, the lone pairs of an oxygen in the ring create an additional hydrogen bond with a hydroxide of the neighboring sugar. And so at the top we have a covalent linkage. At the bottom we have a very strong hydrogen bond. And so we get a lot more integrity in terms of stability and bonding in the cellulose itself. Forms long, straight chains, highly stable because of the hydrogen bonds. It can be degraded by an enzyme called cellulase. We do not produce that enzyme. Rudimentary animals can have some activity. Cellulase by itself is a goal to industrialize. We haven't been able to do that, but if we could use cellulase to break down, let's say, grasses or wood, create individual glucose molecules, which then can be fermented and become a renewable resource. But this has been something that has not been able to be reproduced in an industrial way that is stable enough for us to use, but that is of high interest to a lot of people. Chitin, if you take a look at it, we have a modified glucose molecules Again, these allow us to have not only hydrogen bonds internally, but we can do some other hydrogen bonds. And so the more interaction that you have between these polymers, the stronger the structure can be. And if you think about exoskeletons, that is an armor for the animals. It's a very strong bond. Heteropolysaccharides also can be found in the extracellular matrix. And so here is what an example. If you've ever made gravy, all right, you take the drippings, let's say you cook a turkey and you take the drippings from the turkey and you put them in a pan. But that isn't gravy itself. What has to happen is you have to add something to thicken it up. Most of the time people add either flour or cornstarch, both of which are polysaccharides. And so what happens is when you put the polysaccharide into that liquid, it is so hydrophobic, it will take, so hydrophilic, it will soak up the water and create a gel-like matrix, depending upon how much you have in there. And so you can make a gravy, or if you go too crazy with the cornstarch, you make something that is solid. The gel-like material between the extracellular space in multicellular organisms actually have these polysaccharides that are being dissolved in the liquid and creates this gel-like material that allows adhesion. Now, it is porous enough in terms of water to get through and diffusion of nutrients, oxygen, and small molecules, but they also create that thickness. Most of the time they are fibrous proteins that have a bunch of sugars on them. And so examples of these types of sugars or polysaccharides come in the form of, some of these you may have even heard about, chondroitin sulfate. A lot of times if you've watched commercials and they talk about arthritis or joint pain, if you take chondroitin sulfate, this is a component of the matrix-like material in the joints, and if you are missing that, you can ingest some of these. Hopefully it makes it into your joints, and so it also provides tensile strength for the cartilage, tendons, and ligaments. Keratin sulfate, again, a polymer of, and this is a heteropolymer, these two repeating, found in cornea, cartilage, bone, hair, and nails. And so there's a protein matrix even in the bone where the mineralization occurs, where the calcium phosphate actually bonds to. And so the calcium phosphate will be built on a matrix of this a particular sugar matrix, and we will be able to build the bone, but there is structural integrity. And then inside the eye, it is not truly liquid, it is more a viscous material, and so the synovial fluid and vitreous humor of the eye is made up of this heparin sulfate, which is short polymers of this disaccharide that are dissolved in water and create this really thick kind of gel-like material that is found in those compartments. Most of the time these components have at least a charged residue and quite often have nitrogens in place of hydroxides. The glycoconjugates are those that have the proteins that are either they're proteins that have sugars attached to them, and so again, proteoglycans are mostly carbohydrates with a little bit of protein. The protein is acting as a matrix or a base by which all of the carbohydrates are held. Glycoproteins, on the other hand, is mostly protein with some carbohydrates. The proteoglycans are found on the cell surfaces of the extracellular matrix. and they provide structural support. Whereas the glycoproteins tend to be varied sugars, not polymers, not homopolymers, not disaccharide polymers, but a variety of sugars that are put together in different linkages that are highly branched and provide information by having different three-dimensional structures. Related to that is the glycolipids, which the same type of branching and informational three-dimensional structure is attached to a lipid, so that both the glycoproteins are embedded in the membrane, the lipids are embedded in the membrane, and they are allowing those carbohydrates to be placed onto the outside of the cell. If you take a look at the glycoproteins, we have several different examples of what is called a core sugar. So a core structure is first synthesized if I'm going to make a glycoprotein, of which then we make derivatives of them to create unique individual three-dimensional structures for information. Now, there are two ways in which sugars can be attached to proteins, and that's through the asparagine molecule. And so the asparagine will be part of the protein, and the R group will react with the sugar, and from there the sugar can be branched. The second core is an attachment to a serine or a threonine. So a N-linked sugar or an O-linked sugar are two hydroxide-containing non-aromatic hydroxide-containing amino acids. And so in this case, the core is just a single sugar, and then the branch points are made from there. And you can see there are different colored and shapes. These colors and shapes are defined for what they are, the types of different sugars, and there is a whole lexicon that you can look up and see what they are. I'm not going to go into that kind of detail though for us looking at this. Glycolipids, we attach these, and here is an example of the color coding of what kind of sugar they are. You can see this very large sugar that has some complexity in it being attached to basically a very hydrophobic lipid, in which case this lipid will be inserted into a membrane. Here's the polar head groups to orient it into the membrane, and then the sugar is being presented on the outside. Now, why in the world would we want to place information on the outside of a cell? Well, by having those sugars on the outside of the cell, we can have entities bind to that. And so there is some intrinsic and extrinsic binding. And so you can present the sugar on the outside of one cell. The same cell may present a protein that binds to that sugar pair. And so as you present and you bind, what you do is you create integrity or adhesion between the two cells. And so you can have between cells intercellular adhesion. You can also have some signaling that is intracellular adhesion. Now, a lot of times the body will also present sugars on the outside of a cell for something to bind to it to bring it to the cell. Now, there are a lot of things that are supposed to make an interaction with the sugars that are being presented on this particular cell and brought in so that they can interact in a natural way. Unfortunately, bacterium, and viruses use this mechanism and hijack this mechanism to bind to these sugar entities in the same way an endogenous or a normal binding event should occur. And what happens is, as this binding occurs between these sugars, the bacterium or the virus can invade and can infect the cell. There are quite a number of proteins that are able to proteins and cells that actually use these sugars selectively to bind to these sugars and bring the cell that is needed into the right place. One class is immune cells, and there are other proteins that can bind. And so it is a normal process that is supposed to be there, that is utilized by our body, but viruses, bacterium, and exogenous things have used this interaction to invade the body. The proteins that bind to sugars are called lectins. They bind with high affinity and high specificity to certain three-dimensional shapes. Now, a T lymphocyte, which is part of our immune system, will present sugars, and then a lectin called P-selectin will be the lectin that binds to that particular sugar. Although I did say that bacterium use this method, viruses can use this method, toxins can use this method to make binding that is not natural and hijacks our system to invade our bodies. Now, let's give an example, particular example of the molecular targeting. All right, what we saw in that particular animation was that T lymphocytes, part of our immunity, okay, is being used to interact with, along the blood vessels, selectins, in this case the P-selectin, okay? And what will happen is these particular lymphocytes will bind to fairly weakly to the selectins that are on the membrane of the blood vessels. Now, that association can be broken by blood flow, and the blood flow will push the T lymphocytes down the blood vessels, and what happens is they roll as they go down. And the rolling allows these weak interactions to keep it attached to the blood vessel, but move down the blood vessel itself because there's not a tight interaction. We're going to get out to a signal in which that interaction will become tight, and we're going to have a difference in the function. So if we take a look at this particular leukocyte, as it's rolling down, if there is inflammation or an infection, what's going to happen is signaling molecules are given out. Those signaling molecules, as this rolls in, will increase the binding between the lectin and the sugars to make a strong interaction. That means it stops rolling, and other signaling processes will allow the white cell to move into the inflamed area so that we can have a response. And so the white cells get into the area of infection, and this is where we talk about finding pus, where our body is actually finding the infection. This particular molecular targeting has the interaction of sugars that are presented on the T lymphocyte and the lectin, P-selectin, to have weak interactions most of the time and very strong interactions only when there is indication of an infection or a site of inflammation, which then changes the behavior of what this T lymphocyte will be, or how it will behave. Now, another type of information that can be dictated or seen on the surface of cells is how we do blood typing, the ABO blood typing groups. And so, you go into the doctor's office, they ask, what kind of blood type do you have? I have red blood. Just kind of like, what is your favorite Kool-Aid flavor? My favorite Kool-Aid flavor is red. What we're really doing is we're distinguishing these particular red blood cells by the fact that each one, A, B, and O, on the outside of each of those cells are presenting different kinds of sugars. Now, if you take a look at O, you have a core amount of sugars, the 1, 2, 3, 4, 5 sugars that are in a particular conformation. A has those, so does B have those. The difference is A has one at the end that is different than B, and that is absent in O. And so if you compare these, the body can distinguish between the A, the B, and the O to a greater or lesser extent. And so of these cases, one of these is the general donor, and that is the O antigen, because the bodies of those that have A and B sugars already see this portion of the sugar base. It's just absent one sugar, but having this sugar or that sugar, if you place B blood and an A person, this will be detected as being foreign, the vice versa, and you can have rejection. And it is very dangerous to add blood of the wrong type. Now, why in the world would we have different blood types? Well, it depends upon which glucotransferase that you are expressing, whether you express one, both, or none. And so when sugars are being synthesized, if you have neither the version that is GTA or GTB, then what you will end up being is an O antigen or an O blood type. If by chance you have the gene for the GTA version of a transferase, you'll be moving a certain sugar on the outside of this core to make A-type blood. If you have the other version, the GTB, you might be making the B type, and if by chance you have both versions, you will be an AB blood type. And so it is either the presence or the absence, and this is genetically determined, of the transferases to put those last sugars on the core group of the O. And so, depending upon which transferase you have, you will either have just O, no AB. You can have both A's and B's. You can have just B's, or you can have just A's being presented. If by chance you have A's in your body, then you have antibodies against B structures, so that this antibody doesn't interact with this, so you have no immune response. But if any of this gets in here, this will cause an immune response, which will then break down the red blood cells. Here, the A antibodies will be present in your bloodstream. It doesn't interact with the B, which you are, but if any A gets in there, this will destroy those. If you have AB, then you have no antibodies for A nor B. And if you are a O person, then you have antibodies against both A and B's. And so you can, in this case, for this person who is type A, you can donate to an O or an A. This one can donate to an O or a B. This person can donate to an O, A, B, and an AB. And this person can donate to an O. So you can see that you have O being everywhere. O is the universal donator. One of the last things we're going to take a look at in terms of structural components that are part of the synthesis of sugars is talking about the bacterial cell wall. And we're going to talk about the bacterial cell wall in terms of this is a place in which the use of either lysozyme or penicillin has been used to break down bacterial cell walls in a race to kill bacterial infections. And so one thing that we're going to see is that part of the structural integrity of the cell wall that protects the cell is a polymer of sugars. And it's one of those repeating disaccharide polymers. And so we have two sugars. One is going to be called NAM, NAM, or NAG, NAG, as being the repeating structures. Not only do we have the structure of the sugars that are a polymer that holds things together, but we also have proteins that link the sugar polymers. And so we have basically in one direction sugars holding it together, and then in a second direction we have proteins holding it together, creating a very strong lattice. We're going to use, and we're going to take a look at how penicillin and lysozyme will break either one of the two, whether it be the sugars or the proteins that hold integrity of the bacterial cell walls. Lysozyme, discovered by Alexander Fleming, was discovered in 1922, found in nasal mucus and tears. It's actually part of our defense system, our immunity, in which bacteria can be breathed in, or we can get it in our eye, and this particular enzyme will break down the cell wall, will allow the cells to degrade and to take away their infectiousness by degrading them. The two sugars that we need to know are NAG and NAM, N-acetylglucosamine or N-acetylmuriatic acid. And when we take a look at this, what is the difference? Glucosamine has glucose as its base, and has an amine and an acetyl group. And so that is one. The other is this derivative that also has the N-acetyl, but then it has this very large group that is on the 1, 2, 3, 4th carbon. All right, and what we need to know is the NAG of the two is relatively smaller than the NAM. The NAM is physically bigger. So NAG small, NAM big. So this is small and this is big. That's going to be an important aspect as we move. The lysozyme is an enzyme that breaks down this polymer, and it will break it in a particular place. It'll break it between a NAM and a NAG. And so it must fit into the active site and break at that particular bond, and it will cleave the covalent bond that is holding that together. And so that is important. It breaks between the C1 of a NAM right here and the C4 of a NAG. Now, if I take a look and I feed the lysozyme a substrate, and for simplicity, we are going to use polynag, because polynag will work similarly to the normal substrate, which is NAM, NAG, NAM, NAG, and we use this because it was cheaper, and we could get a lot of this. Now, what we did was we wanted to decide what the structure of the lysozyme's active site and try to figure out what's going on. And so if we gave a polymer of NAG that was 3 residues long, we got 0 activity, meaning it didn't break any of it. When we gave a NAG that had a polymer that had 4 of them, we got a little bit of breaking. When we gave a NAG that was at 5, a length of 5, we got about 60% activity. When we gave a NAG at 6, we ended up having the fastest activity that enzyme could be, and then if we gave it 7, 8, or 9, the activity didn't increase. So if you looked at this experiment right here, how many active sites, or how many, let's say, sugar binding sites, do you expect that lysozyme will have in its active site? Will it have 3, 4, 5, 6, 7, or 8? Well, by the time you get to 6, we have filled the active site. So we know there are 6 sugar binding sites that are in lysozyme, although not all the binding sites are equal. So let's make let's make a chart to talk about what we see. There are 6 binding sites, A, B, C, D, E, and F, okay? This is the laboratory substrate. That was a poly-nag because it was cheaper, okay? And if I fed in a 6-mer or a 6-membered polymer, I got the fastest breaking I could get. I couldn't increase it by having any more length. All right. If this is true, okay, I have 6, and knowing that after analyzing what was broken down, we find that there's a di and a tetramer that was reduced. A di is 2, and a tetramer was 4. All right. So if I break a 6 into 2 and 4, what are the 2 places, theoretically, that I could actually break the covalent bond? There are 2. All right. The first one would be here. That's a possibility. I would break it up into A and B, and then C through F. That's a 2. That's a 2 and a 4. That's not the only place. I could have broken it right here. And then I would have made a 2, a dimer, and then a tetramer if I broke at the secondary place. So I can either break between B and C, and I can break between D and E. I haven't discovered where the breakage occurs, but I have eliminated between E and F, I've eliminated between C and D, and I've eliminated between A and B. I have 2 possibilities. All right. One thing that I did find out later was that D is a little bit too small for the sugars themselves. Okay? And so what I say is that there is a negative interaction when sugar binds in the D site. All right? You think about an active site, every site as a substrate should come down should be a wonderful binding. It should give off energy. It should be a lower energy state. But there was one site that really, it wasn't lower in energy. It actually was higher in energy to put a sugar in this particular position. And so, what was the activity if I fed a trinag into the system? Well, if you go back and you look, a trinag had 0 breakage. Okay. Well, let's look at where trinag could bind. Trinag could bind these 3 spots. Is that a good binding? Well, I've got 2 positives and 1 negative in terms of energy. That doesn't seem great. I could bind here. That's 1, 2, 3. Again, 2 positives and 3 negatives. 2 positives and 1 negative. The third place that I could bind is here. That has 3 very positive interactions. And so the likelihood of trinag to bind into these spots is right here. That's the best binding. Now, that puts a trinag in a position that you could possibly break a sugar. You could break these 2 away from this one. But what did we see with the activity of a trinag? It had 0% breakage. So if this is the most likely place that a trinag would bind, and if this is a possible break point, but we have 0% breakage, that tells us that is not where the breakage occurs. That is the place where the breakage occurs. All right, so we're narrowing down what is happening. All right, now, if I put in the real substrate, remember I told you I had 1 big and 1 small. We said that NAM was big and NAG was small. We also, after some investigation, we found out that site C is relatively small, and it only accommodates one of the sugars positively, meaning only one fits, and that is NAG. So if NAG is there, then knowing the real substrate, this is NAM, this is NAG, This is NAM. This is NAG. This is NAM. All right, and we said that this is where it breaks. If we go back and we said, how does it break? It breaks between the C1 of NAM and the C4 of NAG. And so, if by chance we know that this has to fit here, and this is the break point, it places the NAM and NAG bond right at the place where breakage will occur. That supports all of our experimental data. And so, does everything fit? Yes. Having the orientation where NAG is in the C, followed by NAM, and placing the breakage point here, I will take a 6mer and break it into 2 and a 4, and I'll break it at the right bond. So our experimental conditions have fit our data, and we have identified where the breakage is. Now, we haven't identified why the lysozyme breaks that bond there, but we know that that is where experimentally it should break. So what is the mechanism of this? Okay, well, what we're going to do is we're going to use some, we're going to actually add water across this bond, and so we're going to need to be able to deprotonate and protonate in a particular way so that we can break this bond. And so the catalytic groups that are going to probably be part of this are ones that can deprotonate and protonate, and most of the time they are probably internal to the protein. But remember, proteins themselves like to put the hydrophobics on the inside and the hydrophilics on the outside, and so if by chance you get a polar amino acid on the inside, it most likely is probably going to be part of the active site. After looking at the reactive residues, we find that there's an aspartic acid 52 and a glutamate 35 inside the active site. Their dependence upon reaction has an optimal pH of 5, which is very close to their pKa's, and so this fits experimentally. If we take a look and we look at the polymer, and so I put all of the polymer in here, but I'm just going to call the D and the E bond because that's where we're going to break between. This is what actually happens, okay? Spontaneously inside the enzyme, all right, this bond will break and pick up a proton, and electrons will be now on this particular glutamate. So I've got a negative charge on this side. The aspartic acid is in the negative charge. Here's the polymer part that can float away. Here's the intermediate, and if I break this bond, I form a carbocation. Positive charge that is stabilized by negative and negative charges. So the fact that I have these two residues inside right next to that carbocation tends to make me believe that these can stabilize that. Now this has to diffuse away. The reaction hasn't completed. And what comes in is water. Now this is the negative. It was previously used in an acid, but now it can be used as a base to deprotonate the water. The electrons can attack as a nucleophile a hydroxide at that positive carbocation, and we can hydrate the bond, and now this is the other piece of the sugar, and it can diffuse away. Now, that is a mechanism that has been proposed. The high energy transition state is a carbocation that tends not to be a very stable molecule, but having charge there to stabilize it can help the reaction, although stabilization just because we have charge may not be the only reason why this particular reaction goes. Okay, remember I said that D was the only binding pocket for sugars that actually had a negative, thermodynamically negative interaction, and it's really because there is a steric interaction. So let's talk about that just a moment. The critical catalysis is glutamate first acts as a general acid, and the aspartate in the ionized form stabilizes the carbocation. Now, why is there a bad interaction? Well, the sugars like to be in the chair conformation. Okay, that is great. The problem is, as the D sugar comes in, it physically, sterically bumps up against carbon 1. And so for the sugar to fit in site D, it gets pressed down by the other 5 sugars that are making good interaction. It pressed this one down into a site that is not large enough, and what it does is distorts the structure into what's called a half chair. Now, this carbon 1 in this conformation is a nice tetrahedral that has 109.5 bond angles that is very, very happy. As I move this particular carbon into this structure, well, that's not 109.5 anymore. Okay, this becomes a flat plane. This almost is trying to push it into a trigonal planar conformation. And remember what happens with that bond when it breaks to become the carbocation. Okay. What we do get is a carbon that is in a trigonal planar conformation. So if by chance one of these bonds breaks and picks up a proton, like our mechanism indicated, it will transition from a tetrahedral into a trigonal planar structure, and because of the steric hindrance in this particular enzyme, it accommodates a trigonal planar structure very, very well. This will actually sterically push it towards the structure of the transition state, allowing the reason why a bond will break and pick up a proton, because I'm stabilizing the physical structure of the transition state, along with now the negative charge and negative charge that will stabilize that carbocation. And so that negative steric interaction of binding into that pocket physically moves or transitions the sugar into the transition state structure. The next item, and what happens is every time that you break the integrity of the cell wall, what happens is the bacterial cell has the tendency to break apart because a lot of its stability is within this cell wall. You break that cell wall, you break its integrity. If it breaks apart the cell itself, it loses infectiousness. And so penicillin is another mechanism by which we're going to break the interaction. Now, lysozyme broke the one-directional sugars, and I can break them in multiple places. The penicillin is going to prevent the synthetic development of the other directional stability. Okay, and what we're going to do is we're going to prevent this bit of protein and this bit of protein making that particular peptide bond. Okay, and what will happen is, for integrity, a pentaglycine will be attached to a D-alanine to create that bond, which creates structural integrity. Now, this is one thing. We've seen alanine and glutamine and lysine and all this, but very rarely do we ever see a L or D in front of it, because in almost all places, amino acids are in the L conformation. And here was one of the first times that we saw the D conformation of any amino acid that were in biological systems. And so the D-alanine is very particular. Penicillin, the actual group and structure, okay, the reactive portion is this four-membered ring. This four-membered ring, if you take a look at this, is really strained. because you would want to be in a tetrahedral, you would want to be in a tetrahedral, you would want to be in a tetrahedral. This one you want to be in a trigonal planar, none of which have bonds of 90 degrees. And so if this ring by chance is broken and opens up, it will not reversibly go back. And so if by chance I can react this, break the ring, I get to a place where it is really, really stable. Okay, what we need to know is that that D-alanine and glycine linkage, okay, will be made, but before the glycine attaches to one D-alanine, the cell has two D-alanines that are on the end. The enzyme will bind to the two D-alanines, and the enzyme becomes covalently linked with the carbonyl, and one D-alanine falls away in the intermediate of the synthesis process. So now I have one D-alanine and the enzyme connected. Then the long pentaglycine gets bound, that would go along this way, here's the N-terminus. And what happens is the enzyme facilitates the nitrogen, okay, binding into or attacking the ketone, and the enzyme is the leaving group to create the pentaglycine D-alanine link that creates the stability in the perpendicular direction from the sugars. All right, so what penicillin is going to do is prevent this bond from being formed. Now, how does penicillin stop this bond formation from happening? All right, well, the enzyme has to react with two D-alanines, throw one of the D-alanines off, become linked as part of the reaction, and then bring the glycine in. What's going to happen is we're going to hijack the enzyme. And so what happens is the penicillin will bind to the enzyme that creates that link. It's called glycopeptide transpeptidase. Okay, it's a sugar that has a peptide, and I'm going to make a linkage between the peptides. And so this enzyme wants to bind two D-alanines. Got that? And it will bind to the two D-alanines, react with it, throw one D-alanine, and become covalently linked to the second D-alanine. Well, this binds to two D-alanines, but it also binds to penicillin. If penicillin binds into the active site, the peptidase will react with the penicillin, and I will attack the carbonyl, and I will break that four-membered ring. And what I have done is I have inactivated the enzyme, because now the active site binds to this and it will never let it go. This is a particular inhibitor. It's an irreversible inhibitor. Another terminology that you can use is called it is a suicide inhibitor. This enzyme, although it's perfectly fine in terms of its sequence, cannot do any of the reactions. The cell, the bacteria, would have to create a new transpeptidase for this reaction to be facilitated. Well, why in the world would penicillin bind into the transpeptidase? It's supposed to bind two D-alanines. That's the substrate. Well, take a look. Here's two D-alanines. And if you take a look at this, okay, right here and right here, right here and right here, right there and right there, this atom and that atom, okay, this atom and this atom, this atom and that atom, this atom and this atom, okay. If you take a look at this three-dimensional shape right here and this three-dimensional shape right here, they are very similar. An enzyme is very particular to the substrate that it binds. Not that it knows what the substrate is, it is very particular to a three-dimensional structure. The penicillin binds as well, if not better, than the normal substrate. What happens is it will bind, complete a reaction, and inactivate the enzyme. Whereas if the enzyme reacted with the dealanine, that it's normal, the pentaglycine would come in, displace the enzyme, and it would continue to create stability. The penicillin creates a inactive enzyme, and so less and less linkages can be made when the bacterium is being reproduced, meaning that it's more vulnerable to being broken down and dying away. Now, how in the world do we look at sugars? It's really, really complex because on the outside of cells, we have this informational sugar branching. All right. Quite often, what we can do is we can either break off these particular sugars and separate them either by liquid chromatography, and knowing how fast they run on against a certain column, we can have standards and we can compare them, or we can use mass spectroscopy to look at exact masses and try to identify the sugars by these exact masses. And that's one way to look at it. Another way is to detach the proteins, label them, and then we have antibodies against certain sugars. And so we place the unknown sugars on a whole array of different antibodies. Where the sugars bind, we know the position of the particular antibodies. We know the antibodies and their specificities. And by looking at the grid, we can identify what sugars are there. Or we can use antibodies can bind to them. Lectins are very highly selective binding entities of sugars, and so you can have an array of lectins or antibodies. Again, by knowing the position where they are, you can identify what the sugars are. It is very difficult and a very hard thing to actually identify a brand new sugar actual structure. I remember one of my colleagues that was in grad school, her entire PhD was to identify one unique branching. There's no chemistry like Edmond degradation or DNA sequencing that can liberate one sugar by one sugar. Not only do you need to know the directions of or which sugars they are, but what bonds were formed between them, which is a very complex process. So there are some techniques to do some analysis to actually identify what sugars there are, but this is something that you would study in grad school, or things that you would use if you are studying sugars themselves. I just want you to know there are some techniques, whether antibodies, lectins, or chromatography linked to mass spec, that we can try to characterize what sugars are on cells. All right, that is the conclusion of Chapter 13.